Let's now talk about the overall process of enzyme catalysis, which we saw in the previous lecture, and discuss more in detail how enzymes are regulated. So, enzymes are generally part of a complex metabolic pathways, and their activity is tightly regulated. So, for example, phosphorylase A, which converts glycogen in the liver to glucose 1-phosphate, and thus freeing up glucose from the glycogen storage and supporting metabolism of the cells. So this enzyme is regulated in two ways. First of all, the enzyme requires AMP for the reaction. And at high concentration of AMP, the enzyme is activated, enhances its activity is enhanced. On the other hand, the product of the reaction, glucose 1-phosphate, acts as an inhibitor on the enzyme. And so the product inhibits its own formation, so thus lowering the concentration of glucose in the bloodstream and um, stopping or slowing the release of glycogen from the liver supplies. So this was an example of what we call allosteric regulation, in which the inhibitor binds to a site on the enzyme that is different than the substrate. For this reason, the allosteric inhibitor generally does not look like the substrate. In the case we just discussed, it's a phosphate group that binds to allosteric binding site. Binding to this remote site changes the conformation of the enzyme enough that the active site conformation is also changed and is now unrecognizable by the substrate. So in general, enzymes that are under allosteric control are at the start of biosynthetic pathways. So a biosynthetic pathway involves a series of enzymes all working together to produce a final product. Eventually, though, we will have enough of this final product and we need to slow down its formation. And one good way of doing that is to have the final product of the pathway inhibit the first or second enzyme on the pathway. And this is called a feedback control. Because now the final product does not look like the initial substrate, this inhibition happens at the allosteric site. Another way to regulate the activity of the enzyme is through external stimuli. So we already discussed the action of phosphorylase A, which releases glycogen from the liver reserves and converts it to glucose 1-phosphate. And the activity of phosphorylase A is allowed by phosphorylation. In other words, phosphorylase A is converted to its active form by phosphorylation of the inactive form, which is called phosphorylase B. This phosphorylation happens through a protein kinase, and the protein kinase in turn is activated by signal cascade, which ultimately is triggered by the release of adrenaline in the bloodstream. Adrenaline is a neurotransmitter, which is part of the fight or flight response. In other words, upon release of adrenaline by the surrenal glands, what we have is that we activate a series of metabolic reactions that support, again, muscles mainly. So glycogen is released to support increased muscle metabolism. So in terms of utilizing drugs that act on enzymes, why would we want to act on an enzyme? And generally, as we'll see, we'll act on enzymes by using inhibitors. One example of that is to prevent the loss of a substrate. So, for example, we have that low level of GABA, which is a neurotransmitter in the brain, can cause seizures. GABA undergoes a normal metabolism, and so it's degraded by this enzyme called GABA aminotransferase. What it does is basically carries out a reductive deamination on GABA, so taking out the terminal amine, here the primary amine, and converting this to aldehyde, and by breaking basically the carbon-nitrogen bond. And so this is now inactive. So we have that by inhibiting GABA aminotransferases, we can sustain and support the concentration of GABA in the brain, and therefore that results in anticonvulsant effect. For another example of using an inhibitor will be to prevent the production of metabolite. So for example, gout is basically due to a high concentration of uric acid in the joint, and uric acid can precipitate and form crystals that basically damage the joint by mechanical actions. Uric acid is formed by conversion of xanthine in the presence of oxygen and water to uric acid and peroxide, and also peroxide production so damages the joints. So what we have is we can inhibit the enzymes, xanthine oxidase that converts xanthine to uric acid, and so prevents the formation of this toxic metabolite. Another example is in the cure of infectious diseases. So when we have an infection by bacteria or viruses, we can inhibit enzymes that are essential for the life cycle of the bacterial virus. And the good part of this is that typically those enzymes will be different from endogenous enzymes, and so we can cure the patient without too many side effects, while the effect on the bacterial virus will be to either kill it or to considerably slow its growth and so life cycle and replication. For example, HIV needs to generate viral DNA from a RNA template, and to do that utilizes this reverse transcriptase. So eukaryotic cells, and so human cells, do not utilize this process, and so inhibiting this enzyme acts very well to shut down the replicative cycle of HIV without direct effect on the replication of the patient cell. Another example is penicillin that blocks the biosynthesis of bacterial cell walls. Eukaryotic cells do not have a cell wall, and so utilizing this drug is selective for the bacterial infection and does not damage significantly the host. Another example that we'll see is sulfa drugs that interfere with a synthesis of folic acid by inhibiting this enzyme called dihydropteroid synthase. So humans do not generate their own folic acid. We take it from the diet. And so basically these sulfa drugs have no effect on the concentration of folic acid in the host cells.